I would like to thank organizer for giving me an opportunity to speak here today. Today I'm going to talk about the microbiome analysis we did on different strains of mice with different phenotypes. The main focus of our lab is cancer prevention, and we make a number of mouse models for different projects. And for this particular study, we introduced the same exact gene knockout onto two different strains of mice. And in this talk, I'm going to refer to strain A and strain B. And the gene we knocked out is the enzyme involved in fatty acid metabolism. And both of the knockouts are protected against cancer formation. However, the strain B mice, but not strain A knockout mice, spontaneously develop inflammatory phenotype. So when we open up this strain B knockout mice, we found that mesenteric lymph node, which is a draining lymph node for the colon, was significantly enlarged compared to the wild type counterpart. And when we do the FACS analysis for CD4 positive and CD4 or CD8 positive cells and compare with the spleen, we find that those cell types were significantly increased only in the MLN, but not in the spleen, suggesting that this inflammation is not systemic, but very localized to the colon. So we look at the colon histologically and find up to 20% of the colon is ulcerated. And here is the section of whole man colon stained with H&E, and those arrows are pointing to where the ulcerations are. And if we look closely, this is a normal tissue, colon tissue here. And you can see the colon crypt nicely lining the mucosal layer. However, in the ulceration, these crypt structures are lost, and we see influx of inflammatory cells and fibroblasts. So having these mouse models, one is sensitive and one is resistant to colonic inflammation, we thought that they can represent human populations for sensitivity to mucosal damage, and also susceptibility to the clinical conditions such as inflammatory bowel disease. As part of a characterization of these mice, we examined the gut microbiome because dysbiosis has been linked to the pathogenesis of IBD. For the microbiome analysis, we collected fecal samples from mice over time, before and after the development of colonic ulceration, and we used a strain ID kit from Shoreline Biome to extract and amplify bacterial DNA, which was sequenced by PacBio SQL system. And the results were analyzed by Shoreline Biome using their software. Okay. The unique thing about strain ID kit is that it amplifies a large amplicon. It generates large amplicon using 27F primers and EXT3 primers. And by sequencing this large amplicon containing 16 S gene, internally transcribed spacer, and 23 S genes, we were able to get a greater resolution of strain level variations. So the results are pretty interesting. First of all, we saw a mouse strain specific signature with this analysis. Here's the graph showing the normalized read or abundance, the bacteria on the y-axis, and the strain A is in blue and strain B is showing in red. And as you can see, the species, Akkermansia muciniphila, was significantly decreased in the strain B mice. And the opposite profiles were also seen, where the Bacteroides species were increased in the strain B mice, but the levels of this bacteria remained low in strain A. And we saw more of this profile in the analysis where strain B is missing some bacteria or significantly lower compared to the strain A. And many of those bacteria were butyrate producers, suggesting that, since butyrate is a primary source of energy source for colonic epithelial cells, therefore we suggesting that the strain B mice might have less healthier epithelial cells to start with, therefore it becomes more prone to inflammatory signals. And we are also seeing a group of bacteria associated with the inflammatory phenotype. For example, the species of E. coli was significantly increased in the strain B knockout mice. And we saw many of this pattern, and these bacteria, group of bacteria, was also found in human IBD patient, confirming or suggesting that our mouse model can recapitulate the human IBD cases. And again, the opposite profiles were also found where this bacteria, Prevotella species, were significantly decreased in the strain B knockout mice. And we think those bacteria might have a protective role against inflammation. And the Prevotella species are also interesting when we look at the timeline, where this shows the samples from younger mice to old mice in strain A, and younger to old in strain B mice. And you can see the strain A mice have steady levels of this bacteria. However, in the strain B mice, when they're young, the knockout mice do have this bacteria. However, when they have ulceration or older mice, they lose this bacteria in the system. So all these results opened up a lot of questions for us. And one of them is, do the change in these particular bacteria cause inflammation or vice versa? So now we're currently examining the cause and effects in host microbe interactions by changing the environment. For example, we're co-housing strain A and strain B mice together to see if they exchange any microbiome profiles. And we are also planning to do fecal transplant to supply back those bacteria that's missing in strain B. And also the antibiotic treatment experiments to see if the microbiome is really affecting the inflammatory phenotype in these mice. And most importantly, we wanted to see the levels of bacterial metabolites in the system so that we can see how it's affecting or have impact on signaling pathways. That explains why these mice are having inflammation. Okay, and I would like to thank my boss, Dr. Rosenberg, and students and postdoc in the lab.